A very good evening to all. Today we are going to discuss regarding the application of biotechnology. What is biotechnology? That is the application of technology in living organism for the benefit of the human society. Living organism may be plants, animals, or the microorganism. Founder or father of biotechnology is Carl Ericke, and there are mainly two pioneers in the field of biotechnology. That is mainly two departments. In one is in US, that is Office of Technology. They define biotechnology as any technique that uses living organisms or substances from those organisms to make or modify a product to improve plants or animals or to develop microorganisms for specific uses. And one from UK, that is Interdepartmental Committee on Biotechnology. They define biotechnology as the application of scientific and engineering principle to the processing of materials by biological agent to provide goods and services. Next, application of biotechnology. Mainly we will be discussing regarding the transgenesis and DNA fingerprinting. Transgenesis, that is transfer of the required genes from one organism to another by some methods. So these are main four methods which we are using for transferring required gene from one organism to another, that is from donor to the recipient. The four methods, the first one is retroviral method, second one nuclear transplantation, next DNA microinjection, and the fourth one embryonic stem cell. First two we will be seeing in the coming slides. Regarding DNA microinjection, as the name suggests, the required DNA from the donor is collected and that is injected by using a micropipette through the recipient or the organism or the individual whom to modify. Last method is embryonic stem cells, which states that the at embryonic stage, that is during zygote stage, the embryo undergoes the cleavage and they become the blastocyst. And that stage, the cells are in totipotent condition or pluripotent. That is, each cells are having to becoming a organs or each organs. The germinal layers are not distinguished or they are not determined at that stage. They are known as totipotent cell or stage or the stem cells. They are using for transferring the required gene in the organism. So we will be dealing in the detail. The first method is the retrovirus method. As the name suggests, the retroviruses, they are using as the vector for transferring the required gene from one organism to another. The retroviruses are modified with the required gene or the transgenes are transferred in the retroviruses and they are transferring to the other organism to transfer that required gene. But main limitation is only HGV genes, or we can say that the retroviruses will transmit its own RNA in the organism and that will cause the diseases. As reverse transcriptase is the enzyme in the retroviruses, they cause diseases sometimes. So that is the disadvantage of this method. Next method is the nuclear transplantation method or nuclear transgenesis. That is, nucleus is transferred from the required organism to the recipient to get the character of the required organism, right? The good character or the required character, that is having high intelligence or any specific, like we will be dealing example of a dolly, which is the first cloned mammal. So nuclear transgenic method or nuclear transplantation method. So nucleus is removed from a cell which is transferred or which is fused with enucleated egg cells from the other individual, and they are the nucleus removed cell, that is a nucleated cell, and the nucleus from the donor cells is fused by electric fission, and they will allow to multiply as cycle. So that will give the copying of the required nucleus or the required gene, and that process is known as cloning. And there are mainly two types of cloning: reproductive cloning, that these cloned cells are transferred to in surrogate mother to develop further or to undergo the cleavage. And in the therapeutic cleaning therapeutic cloning, these clones are transferred in the plates, or they are carrying out for the tissue culture. Next, we will be seeing regarding the story of the dolly. As we know, dolly is the first cloned mammal and it's the first cloned sheep. Dolly is a sheep, is the fin dorset type sheep, as dolly got cell or nucleus from the mammary cells of the fin dorset sheep, and they were transferred to the enucleated egg of Scottish blackface sheep, and they were combined by electric fission, and the blastocysts were developed. From the blastocysts developed zygote or the embryos, they were transferred to the surrogate mother or surrogate sheep, other sheep, this is the third one, and they allowed to deliver the eve, that is the dolly. So that is the success story of the dolly, how their Dr. Ian Wilmot and his team in the Roslin Institute, Edinburgh, Scotland, able to successfully produce a mammalian sheep by using the nuclear transgenic method, and this is cloning. He produced successfully able to produce the cloned sheep. In this method, 277 nuclei were transferred, of which only 29 eggs grew into embryo, and of the 29 embryo, 13 only able to transfer to surrogate mother. Of the 13 transferred, only a single eve gave birth to a dolly, and the dolly died after 6 years by osteoporosis, as the dolly is showing the 6 years of character of a mother, Findorset Eve. So that is the story of dolly, the first cloned mammal. The second one application we are going to learn is regarding the DNA fingerprinting, which is also known as the DNA profiling, and which is helping for the DNA sequencing. And the credit goes to the Alec John Jeffrey, who first time able to identify this DNA fingerprinting technique in 1984. As the name suggests, fingerprinting, which vary from individual to individual, this DNA fingerprints also vary from individual to individual. That is, in the DNA, there is a variable nucleotide tandem repeats. They are known as the mini satellite, which are about 10 to 15 base pair length, that is 10 to 15 base pairs, are different from one individual to another, and they are known as variable number of tandem repeats, that is V and TR. Also known as the mini satellites, they are differ from one organism, one individual to another. So DNA will be extracted from the two individuals, and they will be hybridized with the same recombinant DNA enzyme, recombinant polymerase, and PCR is conducted. PCR is polymerase chain reaction, or that will be conducted by using a thermal cycler, and the DNA amount is increased, and after that that will be carried out under southern blotting, and southern blotting is nothing but the DNA. After PCR, they will be running in a gel, electrophoretic gel, or after electrophoresis, we will getting a bands, and they will not be visible, right? So for visible, they will be transferring to nylon. We will a nylon sheet, or we can just transfer it to a nylon sheet, and they will be colored or dyed by probes, mainly lead 32 is using as a probe, in that method is found by the southern, and that is why it is known as the southern blotting, and that will be able to, as the PB32 is a radioactive dye that can be easily recognized by using the X-ray or UV spectroscopy. And in India, Dr. Ralji Singh in CDFT, that is Center for DNA Fingerprinting and Diagnosis, he used a indigenous technique in which the probe which the Alec Jeffrey used, the PB32, that is lead 32, is changed into BKM DNA. BKM is nothing but the banded crate minus satellite. That is a snake. Banded crate is a snake, as Dr. Lalji Singh was doing his study in the banded crate, and he found that the female banded crates, minor satellites, they are the VNTR region, they are similar to the human. So they can be used as the probes to acting as the marker or to identify the differences. So this is the detail in DNA fingerprinting technique. The blood sample of two individuals will be collected. DNA is extracted by standard method, and they are cut into fragments by restriction enzymes, same restriction enzyme. And after that, they will be carried out by PCRs. PCR, after PCR, through increase the amount of DNA, and they are separated. After PCR, they are separated by using the electrophoretic gel, and the electrophoretic gel, they are transfer these electrophoretic cells are transferred to a nylon membrane by a technique known as southern blotting. And in the nylon membrane, they are pouring a radioactive DNA probe, that is the PB32, and the DNA probe will bind with the specific DNA sequences. And excessive DNA probe is washed off. Now they will see under X-ray, and X-ray film is developed, make visible patterns of the bands which are we observed in the previous slide. Like these bands will be observed, and which will be used for the DNA forensic studies, and also for the paternal or maternal disputes, or to identify any specific organism for barcoding or any specific. There are many. This DNA fingerprinting technique has made the revolution in the field of biotechnology. So that is regarding today's class, and these were a few of the sites which you can get the more details about the application of biotechnology, mainly sheep, mainly the first clone sheep, and also regarding the DNA fingerprinting, and also many more. So pictures or the flow charts were taken from these sites. So I acknowledge them for helping me to get the details, or for more reference you can refer in these sites. Thank you all.